Hello, my students. I'm your biology teacher, Cikgu Govins. Our topic today is leaf structure and function. The subtopic is 2.4, main organ for photosynthesis. Learning standard today is, I cover learning standard number 7, 8, and 9. Number 7, explain the environmental factor that affects the rate of photosynthesis, which is light intensity, temperature, and concentration of carbon dioxide. Number 8, conduct experiment to study the effect of environmental factor on the rate of photosynthesis. Number 9, analyze the effect of different light intensity and color of light on the rate of photosynthesis. The environmental factor that affect the rate of photosynthesis, they are carbon dioxide concentration, light intensity, and temperature. Here are the graphs show the relationship between the rate of photosynthesis and carbon dioxide concentrations. The increase in carbon dioxide concentrations increase the photosynthesis rate as long as there is no other limiting factors such as surrounding temperature and light intensity. Limiting factor is a factor that controls a biochemistry process and changes according to other variables. An increase in the limiting factor will increase the rate of certain biochemistry process if the other factors are constant. Okay, this is the graph show this one with high light intensity, this one is with low light intensity. Okay, at P, photosynthesis rate is constant. As the concentration of carbon dioxide increase after P, the rate of photosynthesis remain unchanged because due to light intensity becoming the limiting factor. This is set up of experiments to study the effect of carbon dioxide concentrations on the rate of photosynthesis. Okay. Light intensity. Here are the graphs show the relationship between the rate of photosynthesis and light intensity. Light is needed in the light-dependent reactions. If the concentration of carbon dioxide and temperature are constants, the rate of photosynthesis increases until it reaches its maximum points at noon. Graph 1 show the rate of photosynthesis increases with the increase of light intensity until it reaches a light saturation point at P. After point P, the increase in light intensity from P to Q is no longer increases the rate of photosynthesis because it is limited by other factors such as temperature and carbon dioxide concentrations. Graph 2 show when the concentration of carbon dioxide in the environment is increased to 0.13%, the rate of photosynthesis also increases. This is the setup of the experiment for study the effect of light intensity on the rate of photosynthesis. Now we look at temperature. This is the graph show the relationship between the rate of photosynthesis and temperature. The reaction in photosynthesis are catalyzed by enzymes. Therefore, changes of surrounding temperature will affect enzyme activity and also the rate of photosynthesis. The optimum temperature is different for different plant species, but in general, the optimum temperature is between 25 degrees Celsius to 30 degrees Celsius. A very high temperature denatures the enzyme and the process of photosynthesis is stopped. This is the setup of the experiment for study the effect of temperature on the rate of photosynthesis. Now we look at experiments. how we can conduct experiments in carbon dioxide concentrations, light intensity, and temperature. The problem statement is what are the effects of light intensity, temperature, and concentration of carbon dioxide toward the rate of photosynthesis. Okay, light intensity. The aim is to investigate the effect of light intensity on the rate of photosynthesis. The hypothesis is the higher the light intensity, the higher the rate of photosynthesis. Okay, we look at temperature. The aim also the same, to investigate the effect of temperature on the rate of photosynthesis. The hypothesis, the higher the temperature, the higher the rate of photosynthesis. Carbon dioxide concentrations. To investigate the effect of carbon dioxide concentrations on the rate of photosynthesis, the hypothesis is the higher the carbon dioxide concentrations, the higher the rate of photosynthesis. This is the variables in experiment to study the effect of light intensity on the rate of photosynthesis. Manipulated variable, the distance between the light source and hydrilla. Responding variables, the number of air bubble released in 5 minutes. Constant variables, the type and size of hydrilla, the concentration of sodium hydrogen carbonates, the voltage, the bulbs, the materials, hydrilla, 50 ml of distilled water, 0.2% of sodium hydrogen carbonate solutions. The apparatus is scissor, 60-watt bulbs, meter ruler, stopwatch, paper clip, boiling tube, measuring cylinder, retort stand and clamps, thermometer, and beaker. Here is the diagrams. You can see the apparatus and material. Material is hydrilla, distilled water, and 0.2 sodium hydrogen carbonates. The retort stand, the bulb, the thermometer, and ruler, paper clips. Okay. Temperature. In temperature, the manipulated variable is temperature. Okay. Responding variable is the number of air bubble released in 5 minutes, the same just now. Constant variable, the distance between the light source and hydrilla, the type of and size of hydrilla, the concentration of sodium hydrogen carbonates, and the voltage of the bulbs. This one we need to constant it because we want to study the effect of temperature on the rate of photosynthesis. The material almost the same, only this one we need ice cubes. Okay, apparatus almost the same. Okay, nothing different. So this is the setup of the experiments. You can see here ice cube. Okay, in diagram before that there is no ice cube because this one involved study of temperature on the effect of photosynthesis. Carbon dioxide concentration. This is the variables. Manipulated variable is the concentration of sodium hydrogen carbonate solutions. Responding variable, the number of air bubble released in 5 minutes. Constant variable is the distance of light source and hydrilla, the type and size of hydrilla, the voltage of the bulbs, the materials, hydrilla, distilled water, sodium hydrogen carbonate solution with different concentrations, apparatus, scissor, 60-watt bulbs, meter ruler, stopwatch, paper clip, boiling tube, measuring cylinder, retort stand and clamps, thermometer, and beaker. Light intensity. Here are the procedure. Attach a paper clip under the stem cutting of hydrilla and put them in the boiling tube containing 0.2% of sodium hydrogen carbonate solutions. Clamp the boiling tube vertically to the retort stands. Light the 60-watt bulbs with a distance of 20 cm from the hydrilla. Count and record the number of air bubble released in 5 minutes. Take 3 readings to obtain an average. Replace the 0.2% of sodium hydrogen carbonate solutions in the boiling tube with a new ones. Repeat step 3 to 5 at different distance between the bulb and hydrilla at 30, 40, 50, and 60 cm. Record the result in a table. Here are the results. Distance of the light source when it's 20. The average number of a bubble released in 5 minutes is 20.6. When distance is 30, the average is 17.3. 40, the reading is 11.6. 50, going down to 7.3, the average readings. 60, only 3.6. When we plot the graph, the number of a bubble produced against the distance of light source and hydrilla. Okay, you can see the average number of a bubble decrease, okay, when the distance between the hydrilla and light source is increased, okay, because when the low light intensity, the rate of photosynthesis is decreased. Temperature. Here are the procedure for temperature. Repeat step 1 to 2 as in the experiment in light intensity. Light the 60 watt bulbs with a distance of 10 cm from the hydrilla. Prepare ice water with a temperature of 5 degrees Celsius in a beaker. Count and record the number of a bubble released in 5 minutes. Take 3 readings to obtain an average. Change the 0.2% of sodium hydrogen carbonate solution in the boiling tube with a new ones. Repeat step 3 to 5 for different water temperature, which are 15, 25, 35, 45, and 55 degrees Celsius. Record the result in a table. Here is the result. When temperature is 5 degrees Celsius, the average reading number of a bubble released in 5 minutes is 0. When the temperature is 15, number of a bubble released is 4. 25, this is the highest a bubble release in 5 minutes. 14, 35, the reading going down to 9. When the temperature is 45 degrees Celsius, the average reading is only 2. 55, there is no a bubble release in 5 minutes. So when we plot the number of a bubble produced against the temperature, this is the result of the graph. The highest is 25 degrees Celsius here. Carbon dioxide. Attach a paper clip under the stem cutting of hydrilla and put them in a boiling tube. Pour 5 ml of 0.01 molar sodium hydrogen carbonate solution using a measuring cylinder into the boiling tubes. Clamp the boiling tube vertically to the rattle stands. Light the 60 watt bulbs at a distance of 10 cm from the hydrilla species. Count and record the number of bubble released in 5 minutes. Take 3 readings to obtain an average. Repeat step 2 to 5 for other concentration of sodium hydrogen carbonate solutions. Record the result in a table. So this is the result. 0.01 molar. The average is 10.7, 0.02, 14, A bubble release in 5 minutes. 0.03, 16.3 average readings. 0.04, they are 19.7s. When 0.05 remain the same, and 0.06 also remain the same, the average reading of a bubble release in 5 minutes. So this is the graph. It's constant here, starting from 0.04. Okay. Discussion questions for experiment of light intensity. Based on the plotted graph, state the inference that can be made about the effect of light intensity on the rate of photosynthesis. The answer is, when light intensity increases, more photon will strike the chlorophyll molecule, which will increase the rate of photosynthesis. Question number 2. Explain a method to handle all the variables in the experiments. Manipulate the variables. Fix the distance of the bulbs from hydrilla at 20, 30, 40, 50, and 60 centimeter. That is manipulated variable for experiment of light intensity. Responding variable is count and record the number of a bubble released in 5 minutes. And the constant variable is use the same type and size of plant hydrilla. Discussion question for temperature. Based on the plotted graph, state the inferences that can be made about the effect of temperature on the rate of photosynthesis. The answer is, when temperature increases, the rate of enzyme increases and the rate of photosynthesis also increases. Number 2, explain a method to handle all the variables in the experiments. The manipulated variable is immerse test tube into beaker containing water at 15, 25, 35, 45, and 55 degrees of Celsius. The responding variable is count and record the number of gas bubbles that can be released in 5 minutes using a stopwatch. Constant variable, use the same type and size of plants. Hydrilla. Discussion question for carbon dioxide concentrations. Based on the plotted graph, state the inferences that can be made about the effect of concentration of sodium hydrogen carbonate solutions. The answer is, when the concentration of sodium carbonate increase, the rate of photosynthesis will increase because of higher concentration of carbon dioxide. Number 2, explain a method to handle all the variables in the experiments. For this one, manipulated variable is using sodium hydrogen carbonate solution at 0.01, 0.02, 0.03, 0.04, and 0.05 molar. Responding variable, count and number of a bubble released in 5 minutes. Constant variable is use the same type and size of plant, hydrilla. Now we see the effect of different light intensity and light color on the rate of photosynthesis. What is the best light color to maximize the rate of photosynthesis? The answer is red and blue lights. The wavelength range for red is 600 to 700 and the blue light is 425 until 450. The rate of photosynthesis in plant is different throughout the day. This is because all the red light is absorbed by chlorophyll. The blue light is absorbed by carotenoid pigments before being transferred to the chlorophyll. These two lights have enough amount of energy to excite electrons in the light-dependent reactions. Now we look at formative practice 2.4. Here are the questions. State 3 main factors affecting the rate of photosynthesis. Number 2, state the effect of different light intensity on the rate of photosynthesis. Number 3, Mr. Kumar has succeeded in planting grape plants in a greenhouse in Cameron Highlands. List the equipment needed. Number 4, light-independent reactions depend on light-dependent reaction. Do you agree with these statements? Explains. State 3 main factors affecting the rate of photosynthesis. The answer is light intensity, temperature, and carbon dioxide concentrations. Question number 2. State the effect of different light intensity on the rate of photosynthesis. At low light intensity, the stomata will close and lower the rate of photosynthesis. At high light intensity, the stomata will open and until it reaches a point. After the points, the rate of photosynthesis does not increase because other factors become limiting factor. Number 3. Mr. Kumar has succeeded in planting a grape plants in a greenhouse in Cameron Highlands. List the equipment needed. The answer is supply of water, bulb with appropriate light intensity, adjustable heater at 25 degrees Celsius, optimum temperature for enzyme reactions in photosynthesis, and carbon dioxide source. Light-independent reactions depend on light-dependent reactions. Do you agree with these statements? Explains? Agree. A light-independent reaction requires an ADPH and ATP, which is produced in light-dependent reaction. That's all for today. See you in next lessons. Thank you. Bye-bye.